A bird crosses the Sahara at night using a sense organ that runs on a single unpaired electron. A plant converts sunlight into chemical energy at 95% efficiency by letting a photon exist in multiple places in the leaf at once. A fruit fly can tell the difference between two molecules that are chemically identical, atom for atom, mass for mass, and the only thing different between them is the way their nuclei vibrate, which is a quantum property, not a chemical one. Three completely different branches of biology, three completely different noble adjacent labs, all converging on the same uncomfortable fact. Life uses quantum mechanics, actively, efficiently, at body temperature, which means the argument you've probably heard, the one that sounds so satisfying, so scientifically mature, so appropriately skeptical, that the brain is too warm and wet and noisy for quantum effects to survive. That argument was already wrong before anyone finished saying it out loud. I want to be precise about what I am and am not telling you tonight, because this topic has a way of making people either believe too much or dismiss too fast, and both of those are failures of the same muscle. I am not telling you that your soul is a qubit. I am not telling you that consciousness has been explained. What I am telling you is that the single most common reason educated people give for refusing to even entertain a quantum theory of the mind is a piece of physics that stopped being true around 2007, and almost nobody got the memo, because the correction happened in a photosynthesis lab and not in a philosophy department, and those two buildings don't email each other. Here is the belief, stated the way a smart, science-literate person actually holds it, because I want you to recognize it in your own head. Quantum superposition, a particle existing in multiple states at once, is a delicate, cold, laboratory phenomenon. It requires temperatures near absolute zero, or a vacuum, or extreme isolation, because the moment a quantum system touches its warm, chaotic environment, it decoheres. It collapses into ordinary classical behavior. In a timescale so short, it might as well be instantaneous. A human brain, at 98.6 degrees, soaked in ions and water and constant electrochemical noise, is about as far from a quantum computer's operating conditions as you can physically get. Therefore, any theory that puts quantum mechanics inside a neuron is not cutting edge, it's crackpot. That is the belief. It is not stupid. It is, in fact, exactly what the physics of the 1990s would have predicted. The problem is that biology didn't wait for physics to update its priors. Before I go further, I need to give you one piece of physics, vocabulary, and I'm going to do it the way I'd do it for a room of biologists who never took a quantum course, because most of this audience didn't either, and the argument falls apart if you're nodding along without actually holding the concept. Superposition does not mean we don't know which state it's in. It means the system is, in a mathematically precise and experimentally verified sense, doing both things at once and only resolves into one outcome when it's measured. When it interacts strongly enough with something else that the information about which state it's in leaks out into the environment. That leaking is decoherence. It's not mysticism. It's not the observer's consciousness collapsing a wave function the way pop culture likes to claim. It's a bookkeeping process described precisely by Wojciech-Jurek's Einselection framework in the 1980s and 90s, where environmental interaction picks out which quantum states survive and become effectively classical, and how fast that happens depends entirely on how strongly and how noisily the system is coupled to its surroundings. The 1990s consensus was built on Jurek's math applied to simple systems, a single particle in a gas, a photon in open air. And in those systems, warm and noisy really does mean fast decoherence, often in femtoseconds. The mistake, and I want to be exact about whose mistake this was, because it wasn't a stupid one, was assuming that every biological system would behave like those simple textbook cases, without anyone actually running the experiment on a real protein doing real biological work. Let's go to the mechanism, because I promised you gears, not vibes. In 2007, a team led by Graham Fleming at Berkeley shot ultrafast laser pulses at the photosynthetic light harvesting complex of a green sulfur bacterium and watched, in femtosecond time resolution, an energy packet, an exciton, if you want the word, travel from the point where it absorbed a photon to the reaction center where that energy actually gets converted into usable chemical fuel. Classically, that energy should hop randomly from one chlorophyll molecule to the next, like a drunk man bouncing off street lamps, taking a wandering, lossy, inefficient path. Instead, the data showed oscillating quantum beats. The signature of the excitant traveling as a coherent superposition, sampling multiple paths through the molecular scaffold simultaneously, and effectively selecting the most efficient route the way water finds the lowest point in a landscape without checking every possible stream first. This is called quantum coherence in a warm biological system, and it should not exist according to the decoherence math everyone was using. It existed anyway. The bacterium had, through billions of years of evolution, apparently built molecular scaffolding, protein vibrations tuned to specific frequencies, that protects the coherence from the very noise that was supposed to destroy it in femtoseconds. Noise wasn't just failing to kill the quantum effect. In some models, the noise was structured in a way that helped sustain it. That is the gear that breaks in the old argument. Warm and noisy was never the real killer of quantum coherence. Untuned, structureless noise is the killer. Life had apparently found a way to structure it. I opened a loop up there, and I'm going to leave it open for a minute, because before I close it, I need you to understand where it goes next, and that requires the second piece of evidence, which is stranger than the first. In 2011, at Oxford, a physicist named Eric Gauger and a group including Vlatko Vedral proposed a mechanism for the European robin's magnetic compass, the sense that lets a bird weighing less than a can of soda navigate thousands of miles using Earth's magnetic field, which is roughly 100 times weaker than the field from a refrigerator magnet. The leading model, called the radical pair mechanism, works like this. A photon hits a protein called cryptochrome in the bird's retina, and that photon kicks an electron loose, creating a pair of molecules each carrying one unpaired electron. Those two electrons are quantum entangled. Their spins are correlated in a way that has no classical analog, and Earth's magnetic field, weak as it is, subtly influences how those entangled spins evolve over time, which changes the chemical products the reaction produces, which the bird's visual system apparently reads out as a pattern overlaid on what it sees. The entanglement in this system has been estimated, in follow-up experimental work, to survive for tens of microseconds inside actual bird retinal tissue, orders of magnitude longer than physicists in the 1990s would have permitted, in a system that is wet, warm, and about as far from a dilution refrigerator as you can get. A robin is running a quantum sensing experiment in its eyeball to figure out which way is south, and it is more sensitive than any magnetometer humans had built at that scale, until quite recently. Now I'll close that loop from a minute ago and open the next one at the same time, because that's how the good arguments in science actually chain together. Every closed door is the hinge of the next one. The loop was, if untuned noise doesn't automatically kill coherence, what does protect it? And does the answer stop at plants and birds, or does it go further? In 2011, a group in Berlin led by F. Stratios Manisakis, followed by more detailed olfaction work from Luca Turin and later computational modeling by Marshall Stoneham, revived and refined a theory of smell that had been dismissed for decades. That your nose might not be a simple lock-and-key shape detector, matching odor molecules to receptor shapes the way a key fits a lock, but might instead, at least partially, detect the quantum mechanical vibration frequency of molecular bonds through a process called inelastic electron tunneling. The electron doesn't just sit there, it tunnels through a barrier it classically shouldn't be able to cross, and in doing so it loses energy, matching the exact vibrational frequency of the bond it tunneled past. The evidence that tipped this from fringe to seriously testable: molecules that are nearly identical in shape but differ in isotope, meaning the atoms have different masses and therefore vibrate at different frequencies while occupying identical geometric space. Smell different to fruit flies, trained to discriminate them, and in some controlled human trials, to people. Shape-only models have no mechanism to explain that. Vibration-based models predict it directly. This is not settled science. Turin's theory has real critics and real unresolved experimental gaps, and I'll come back to that, because pretending otherwise would make me exactly the kind of scientist I'm here to argue against. But the mere fact that it's a live, funded, published controversy, rather than something laughed out of a physics department on arrival, the way it would have been in 1985, is itself the point. The immune system to quantum effects in biology that used to exist in the scientific community has been substantially disabled by better instruments. That's not a metaphor. That's a documented 3-decade shift in what counts as a serious hypothesis. And most people who consider themselves scientifically literate are still running the old antivirus software. I said I'd come back to this 5 times, and I mean to keep that promise. So let's return to the persistent opponent directly. The wrong idea is, decoherence in warm systems is fast and total. Therefore, quantum effects are biologically irrelevant beyond the trivial chemistry everyone already accepted. Therefore, any theory connecting quantum mechanics to the brain is pseudoscience by default. I want you to notice the structure of that sentence, because it has a hidden move in it. It goes from a true premise. Decoherence is real and fast in most systems, to a false universal. Therefore, it's fast and total in all biological systems, without checking. Photosynthesis checked, magnetoception checked, olfaction is currently checking. The brain has not been checked nearly as thoroughly, and the people most confident it will come back negative are, disproportionately, people who have not looked at what happened in the other 3 fields. I promised the opponent 5 direct hits, and we've only landed 3. So let's take the 4th from a completely different corner of biology. One that doesn't even require light or magnetism. Just an enzyme doing the most boring job in the cell. Enzymes speed up chemical reactions by factors that can reach 10 million or more compared to the same reaction happening in open solution. And for decades, the explanation was purely classical. The enzyme shape squeezes the reacting molecules into the correct position and lowers the energy barrier they have to climb over. That's true, but it's not the whole story. And the part it leaves out was nailed down by work from Judith Klinman's lab at Berkeley on an enzyme called alcohol dehydrogenase, the same one your liver uses to break down ethanol. When you swap a hydrogen atom in the reacting molecule for its heavier isotope, deuterium, a purely classical climb over the barrier model predicts a certain slowdown in reaction rate, because deuterium is heavier and should take longer to get over the hill. The actual slowdown measured in the lab was far larger than that classical prediction, sometimes by a factor of 10 or more beyond what climbing the barrier alone should cost, and showed a temperature dependence that classical over-the-barrier chemistry does not produce. The explanation that fit the data was that the hydrogen isn't climbing over the energy barrier at all. It's tunneling straight through it. Quantum mechanically passing through a wall, it doesn't have the classical energy to climb. The same phenomenon that lets electrons tunnel through the barrier in your nose's odor receptors, the same phenomenon that lets particles escape the gravitational pull of a black hole through Hawking radiation. Your liver, right now, breaking down whatever you had to drink this week, is running a quantum tunneling event billions of times a second at 98.6 degrees, in a system so wet it's mostly water. That's 4. Photosynthesis, magnetoreception, enzyme catalysis, and olfaction under active investigation. 4 separate corners of biology, 4 separate noble track labs, none of them set out to prove a point about consciousness, and all 4 of them independently discovered that the too warm and wet argument has a much smaller jurisdiction than the people repeating it assume. Let me put a number in front of you that I chose specifically because it should make you do arithmetic in your head and not like the answer. The 2011 magnetoreception coherence times I mentioned, tens of microseconds, sound tiny. Here's the comparison that should bother you. The best superconducting quantum computer qubits that IBM and Google were building around that same era, held inside dilution refrigerators, cooled to nearly absolute zero, wrapped in vacuum chambers, shielded from every stray electromagnetic field engineers could think to shield against, were achieving coherence times in a similar range, tens to low hundreds of microseconds, using $1 billion of infrastructure to fight decoherence. A bird's retina was matching that performance at body temperature in daylight, while the bird was actively flying, using a protein it grew from an egg. That's the number. Sit with it. A house sparrow's eyeball is competitive with a cryogenic engineering marvel, doing a version of the same fundamental physics. If that doesn't rearrange your intuitions about what warm and messy rules out, I don't think a bigger number would have done it either. Sometimes the comparison itself is the argument. There's a detail in the magnetoreception story I deliberately held back, because it's the kind of thing that should keep you up at night in a good way, and I want it sitting by itself for a second before we move on. The radical pair mechanism doesn't just require entanglement to exist. It requires that entanglement to be sensitive to a magnetic field roughly 100 times weaker than the field coming off a refrigerator magnet on your kitchen door. And it requires the bird's nervous system to read out the tiny chemical difference that field produces as if it were a visual overlay. Some researchers describe robins potentially perceiving it as a kind of shading or pattern superimposed. On ordinary sight, though nobody, understandably, has managed to ask a bird what that actually looks like from the inside, which means the sensitivity isn't just present, it's tuned to a threshold that sits right at the edge of what quantum mechanics allows and classical chemistry forbids, in a way that would be an almost absurd coincidence if the underlying mechanism weren't quantum. That's the open loop. Why would a classical chemical process happen to be tuned to exactly the sensitivity level that only makes sense if something quantum mechanical were doing the sensing? Here's the close. It wouldn't, and that's precisely why the leading researchers in this specific field, including physicists who started out deeply skeptical of quantum biology as a phrase, have shifted their public position over the last 15 years from "this is an interesting curiosity" to "this appears to be the only mechanism that fits the sensitivity data we've actually measured." That shift in expert consensus inside one specific well-funded, heavily scrutinized subfield is the closest thing biology has to a controlled experiment on whether the warm systems can't sustain useful quantum coherence assumption survives contact with better instruments. It didn't survive. And that's the foundation the next part of tonight's argument stands on. Because if it can happen in an eyeball built from ordinary embryonic development, the question of whether something structurally similar could be happening inside 3 pounds of far more complex, far more highly evolved neural tissue stops being a question you're allowed to dismiss with a shrug and a temperature reading. I need to tell you something about myself here, because you should know exactly how far my confidence goes and exactly where it stops. And I'd rather you hear that from me than discover it by catching me overselling something later. I spent years assuming, the way most working physicists and biologists assumed through the 80s and 90s, that decoherence theory had basically closed this door, that Max Tegmark's back-of-envelope 2000 calculation, which estimated decoherence times in neural microtubules on the order of 10 to the minus 13 seconds, was close enough to the final word that further inquiry here was mostly a waste of a career. Tegmark's calculation was a real calculation, done by a real physicist, and it wasn't wrong about the specific system he modeled. What unsettles me, genuinely, sitting here now, is that the photosynthesis and magnetoreception results came after that calculation and did not fit the intuition it produced in the field, including in me. I don't currently have a clean answer for why biological systems seem able to engineer around decoherence timescales in some contexts and, as far as anyone has shown convincingly, apparently not in others, including, and this is the part I can't close, inside an actual living human neuron doing actual human thinking. Nobody has run the Fleming-style femtosecond experiment on a functioning cortical microtubule in a living brain and gotten a clean coherent signal back. Stuart Hameroff and Roger Penrose have a specific, falsifiable proposal for where to look. Microtubules, the hollow protein tubes that form part of a neuron's internal skeleton, and tubulin, the protein those tubes are built from. And here is the piece of evidence that keeps this from being pure speculation, the piece that unsettled me most when I actually sat with it: anesthetic gases. Halothane, isoflurane, xenon. Chemically about as different from one another as molecules get, some of them containing no carbon at all, xenon being nothing but a single noble gas atom. All reliably switch off consciousness in humans at remarkably precise, reproducible concentrations, while leaving basic neural firing, reflexes, and even a fair amount of ordinary brain electrical activity comparatively intact. A purely classical, consciousness is just the pattern of neurons' firing model, has a genuinely hard time explaining why chemically unrelated molecules, sharing almost no obvious structural feature, all hit the same target with that kind of precision, unless what they're actually disrupting is something more specific and more fundamental than firing patterns, something like the delicate quantum vibrational, States inside tubulin that Hameroff and Penrose's model depends on, which recent work using terahertz spectroscopy on tubulin lattices in 2014 and again in more refined form in 2022 has shown do support collective vibrational resonances at exactly the frequency ranges the theory predicted, resonances that are measurably altered by the same anesthetics that switch off human consciousness. That is real data. It is not proof that or is correct, but it is the reason a theory that looked, 20 years ago, like a science fiction writer's daydream. And I say that as someone who has genuine respect for what science fiction writers are allowed to get away with, is still being published in serious physics and neuroscience journals instead of being buried the way cold fusion was buried. Falsifiable and confirmed are different words, though, and I will not pretend the gap between them has closed just because the story got a good chapter. Nobody has run the Fleming-style femtosecond coherence experiment on a functioning cortical microtubule inside a living, thinking human brain, and pulled a clean, unambiguous coherence signal back out. That's the crack. I'm not comfortable in it. I don't think anyone honest is, and I'd trust this story a great deal less if somebody stood up here and told you otherwise. And that brings the opponent back one more time, because this is the moment it usually resurfaces loudest. Somebody in the comments under this very video is going to write that anesthetic evidence, tunneling evidence, coherence evidence, all of it, is just clever pattern matching, the same kind of coincidence hunting that finds Jesus in a piece of toast, and that real scientists moved past quantum brain nonsense decades ago. I understand the impulse behind that comment, and I want to be fair to it. Skepticism of an exciting-sounding theory is usually the correct default setting, and it has protected science from an enormous amount of nonsense over the centuries. But notice what that comment is actually doing. It isn't citing a specific experiment that ruled the mechanism out. It's citing a vibe. The same warm and wet vibe we started with tonight, dressed up as if it were still current, when 4 separate fields of biology have spent the last 2 decades quietly proving that vibe wrong in every other context where anyone actually bothered to check. So where does the floor actually drop? Because I told you it would, and I don't renege on that kind of promise. Here it is, if it's true. And I want that if in bold, in your head, permanently. If the specific mechanism Penrose and Hameroff proposed, or something structurally like it, turns out to be how consciousness actually operates, then the thing sitting at the base of human experience is not merely electrochemical, the way a thermostat or a calculator is electrochemical. It's something closer to what's happening inside that robin's eye and that bacterium's photosynthetic membrane. Matter organizing itself to protect and exploit a physical resource. Coherent superposition. That the rest of the universe is actively trying to destroy at every instant, everywhere, all the time, through the ordinary thermodynamic tendency toward disorder we call decoherence. Which means consciousness, if this is right, is not a passive readout of neurons firing in sequence like dominoes. It's an ongoing, continuous fight against the second law of thermodynamics, waged at the molecular scale, inside you, every second you are awake enough to be having the experience of reading these words. You are not a machine that happens to think. You are, on this picture, a standing wave of resistance against entropy's most basic instruction to the universe, and the moment that resistance stops, the moment decoherence wins completely and permanently, is the moment usually called death. I want to push on that a little further, because it's easy to let a sentence like that wash over you as poetry and move on. And I told you at the start I wasn't going to let myself get away with that, so I'm not going to let you get away with it either. Every other physical system we know of loses its battle with decoherence essentially instantly and never notices, because nothing in that system depends on winning. A rock does not care that its quantum states collapse into classical ones in some unimaginably short fraction of a second, because a rock isn't using superposition for anything. What the photosynthesis experiment, the magnetoreception experiment, and the enzyme tunneling experiment all show, independently, is that when a biological structure does have a use for a fragile quantum resource, evolution appears capable of engineering scaffolding, precisely tuned protein vibrations, precisely shaped molecular cages, that measurably slows the rate at which that resource is lost, not stops it, slows it, buys time with it, extracts work from it before the environment wins. If the same trick that plants use to move energy and birds use to sense magnetic fields is also, in some more elaborate and currently unconfirmed form, the trick your brain uses to generate the experience of being a self that is reading this sentence right now, then what you call you is not a noun. It is a verb. It is an active, ongoing, energetically expensive process of holding a fragile physical resource together against a universe that is, at every single instant, trying to tear it apart and average it out into the same undifferentiated thermal noise that everything else eventually becomes. Every thought you have ever had on this picture was a small, local, temporary victory purchased at a real physical cost, and every night you fall asleep is a partial retreat from that fight, and every death, everywhere, at every scale, of every conscious thing that has ever lived, is simply what happens when the universe finally collects on a debt it started charging interest on the moment that structure first assembled itself. That is a genuinely bigger question than is the brain quantum? That is experience itself a physical process that is, at bottom, a local, temporary victory against the thing the universe is doing to everything else, all the time, without exception? I don't know if that's true. I know it's the kind of question that makes the original argument. Brains are too warm for quantum effects. Look like it was never really about physics at all. It was about how uncomfortable a species gets when its own inner life turns out to be stranger than its metaphors for it. The birds already knew the trick. The bacteria already knew the trick. The only organism that seems embarrassed by the possibility that it might be running on the same trick is the one writing papers about it. And I think that tells you something about the difference between intelligence and self-image. And it is not a flattering difference. And it is the last thing I am going to say to you tonight. So take it exactly as bluntly as I mean it. The brain was never too warm for quantum mechanics. It was only too proud to check.